Alzheimer's disease. Alzheimer's disease very rarely arises from genetic mutations that are transmitted in families. Only about 1% of the Alzheimer's arises in that way, but they result in an early onset form of Alzheimer's disease. Normally, it doesn't begin to affect patients until age 60 or 65, but in these early onset cases, it can be as early as age 30. The mutated genes encode proteins that directly produce A beta, APP secretase, and gamma secretase, as shown on the last slide. And all mutations result in increases in A beta levels in the brain, particularly the A beta 42 form. I'm sorry about these technical difficulties.
Oh, no problem. I'm seeing the preview.
I'm maneuvering my mouse over my screen, but I can't get full screen back here.
Is PowerPoint maybe open?
Now that got it up on my screen, but is it on yours as the full size?
Not full screen, not full size on my end anyway. Oh, perfect. It's full screen now. Thank
Okay. I had already gone to the next slide right here, but this part that's partly obscured in my screen, and
Yeah, I feel like Zoom puts their buttons in the most inconvenient spots. I see it full screen now.
okay, thank you. Well, I'll just stay away from those half-open circles that I don't know. And I got one circle that will advance the slides, so I'll try to work with just those. I kind of skipped over this slide before hitting the genetics text slide. This is a membrane-bound protein, the APP, which is the precursor of A beta that you see on the left side of the screen. And there are two processing pathways that are shown, but I'd like to emphasize the top pathway that results in A beta being formed by two protease clips, the beta and the gamma proteases. It's shown as being released from the membrane, but that's really a topic that I'm uncertain of, and in fact, I'm beginning to make progress on what happens if not all the A beta is released from the membrane or not. And this is the slide that I've already shown. Now, this is what happens to A beta when it sits in cells for a long period of time. It forms a double-stranded beta sheet, and two beta sheets associate with each other. And this is kind of a cross-section of something that proceeds into the screen by several nanometers. It's literally thousands of A beta sheets, and they form a fibril that can be seen histologically in slides of the brains of people who've died of Alzheimer's disease. But as research in A beta progressed over the years, people began to wonder whether the fibrils were in fact the infectious form. And today that's still an open question. There are half a dozen different types of A beta fibrils, A beta 40 and 42 fibrils in particular. They're 40 amino acids long or 42 amino acids long. But there are dozens of oligomers, which are small assemblies, anywhere from aggregates of 2 peptides up to roughly 100 peptides. And they're formed in various tissues in various ways. Unfortunately, they're all very small in proportion, and we can't isolate from brains enough of any one oligomer to characterize it. So we've been synthesizing A beta in the lab and subjecting it to various conditions that allow it to aggregate in these various ways, and we can get information. For example, this structure here was a fibril that was generated in vitro by particular kinds of conditions. What we did is to isolate the oligomers by SEC. A beta 42 undergoes initial aggregation in dilute SDS, which forms micelles, and those are thought to be a reasonable model of biological membrane. Then we dialyze out the SDS and apply them to an SEC column, size exclusion chromatography column, that is called SuperDex 75. And we run the chromatogram. It separates peaks by size, with the larger materials coming out first and the smaller material coming out second, as you see in the panel A here. In addition to that, we also ran well, I can't remember the name of it now, but it gives the molecular weights of the various portions, and the molecular weights are shown here in the dashed line. It appears as a thicker line than the SEC molecular absorbance units, and it tends to level out at a molecular weight of about 150 kilodaltons. So in the conclusion of that first text on the left was nearly constant across the early peak. And then we did size fractions across the peak. Those are shown in panel B. And the gel was stained with silver. That stains protein of any kind that you fractionate on an SDS polyacrylamide gel. And the distribution was identical across fractions 21 to 27, indicating partial dissociation of the relatively homogeneous 150 kilodalton oligomer. Am I keeping you with me?
Yes, yes.
Okay.
Yes.
We then conducted solid state NMR over a couple of years, looking at various heavy labeled amino acid forms of A beta 42. And the NMR gives us distances between the residues that are labeled. And from that, we were able to deduce the following structure. You see the N strand on the left and the C strand on the right. The C strand is just the final 12 amino acids of the A beta 42 molecule. It's a 42 amino acid peptide. And we immediately identified it as an beta strands indicating an antiparallel organization. And that's indicated in the red arrows pointing in opposite directions. The N strand, which we tried labeling in various ways, similar to what we'd done with the C strand, we didn't get any anti-parallel N strands. And we finally conducted enough NMR experiments that we deduced that it was actually a parallel beta sheet strand, but it was out of phase by 3 residues in alternating strands. So that was an unusual configuration to have both parallel and anti-parallel beta structure in the same peptides. But then we were able to do a lot of molecular modeling, and we also did some low resolution cryo-EM of these 150 colonal oligomers. The cryo-EM showed a constellation of 4 subunits, four oligomers that was in a planar arrangement. And we did some class averages and simulated our oligomer model with the format shown on the right, which is a relatively complicated formula. The C strands are shown in red, the N strands are shown in blue, and the N strands are alternatively L and U shaped. And that's sufficient to make an electron micrograph that is sort of in the arrangement that we see here in cryo-EM. So that's the kind of research that I do. It's very biochemically and biophysically based, as you can see. And Gabby has made a very good choice of having me talk about the psychology of the brain. And I have gone through the slides, but I know a little more than you do, and that's about all. I'm going to stop sharing now, and we'll have Jordan put the screen up with the slides that we want to view today.
Before we move on, does anyone have any questions for Dr. Rosenberry about the slides he presented or about his work at Mayo?
Yeah, I just wanted to clarify a comment that you mentioned. You said only, did I understand correctly, 1% of all Alzheimer's cases are genetic? Or is it just for the early onset?
That's for all forms of the disease. Most of the disease is labeled as sporadic, namely it arises in the general population without any family history. However, a number of investigators over several years have identified additional genes that may have an effect on developing sporadic Alzheimer's disease, in particular those that have one form of the HDL protein, which is called ApoE, and it's ApoE4, ApoE3, or ApoE2. About 10% of the population has ApoE4, and that's a risk factor for Alzheimer's disease.
Oh, wow. Okay. And we're able to test these certain proteins while
Yes.
people are still alive? Okay, wow,
Yep.
nice. Interesting, thank you.
Okay.
I have the slides up. Can everyone see them?
Yes.
Yes.
All right, then we are good to go.
Let's have the first slide. Okay. Perception and interpretation. Chapter 5 is basically on the way our sensory fibers and the reception of the receptors in those various senses of sight, hearing, smell, and so forth, are relayed in the brain. And what's clear to me after reading the chapter, and also from general experience, is that the initial fibers that receive the information from the receptors, visual and hearing receptors, for example, activate multiple neurons as they enter the brain. And the interpretation of that signal gets incorporated into the fibers and into the brain itself. The next slide. And that's called the interpretation part of it. Perception occurs as the first filter. Interpretation of these perceptions through comparing them to memories of past experiences and observations occurs. So you classify the information that you receive, and you're not even really aware of this, according to past experiences. Next slide. Next slide. Short-term memory, which you've, I guess, heard a little bit about in previous chapters, is a limited phenomena. It's only stays with you for at most a few days, but it allows the brain to build simple representations of people, places, objects, and so forth as references. And then the brain makes sense of moment-to-moment perceptions through a complex network of associations assembled from prior experiences. Example, you can see it, recognize a dog as being a dog, even if it is a different breed than you've ever seen before. Next. The short-term memories are incorporated into somatic memory. It's a declarative form of long-term memory for facts and data, and it is reinforced by signals that continually enter into that field and make them strong enough to become long-term memory. It's also necessary for reconstruction of long-term representations initially interpreted by short-term memory. Next. Those neurons project all over the brain. They're found in various tissues that are observed to be specific for certain phenomena. Specific cortical networks are specialized for processing certain types of information, and the regions of the cortex selectively process different categories of information, including animals, faces, tools, or words. So this, your perceptions are translated into a relationship of neurons that may be thousands of neurons involved. And each one is a very specific information that forms a long-term memory. And when a new electron passes down the initial neuron that's receiving the signal, if it forms a close representation to that memory, it will be integrated in with that memory and stored in long-term memory. Next, please. The electrical activity of individual brain cells is low, but single cells may fire when someone looks at a particular person, but not when viewing general picture of people, animals, or objects. And that's because the single cell firing results in a cascade that is amplified throughout the brain. And if it doesn't quite match the people, animals, or objects that are stored under similar representations, it doesn't fire as often. Concept cells work together in assemblies, and they reside in the temporal lobe. That's the blue area of the slide, which is important for object recognition. Cells encoding the concepts of needles, threads, sewing, and buttons may be interconnected, meaning that they have several of integrated neurons that are aligned, but they're still different enough you can recognize the individual concepts. Next. The way this information was obtained was to look at particular patients, and there are 3 or 4 cited in the text that you have. They're generally not as specific as the ones that are outlined. Patient DBO, for example, was damaged by a stroke in the temporal lobe, prevented visual input from being conveyed to the anterior temporal regions where somatic processing occurs. And that was where the stroke occurred, in the temporal lobe, and it blocked his access to names of objects that he could see, but not his ability to name objects he could touch. So his sensory neurons that involved in touch were fully intact, but his ones associated with this visual ability were interrupted. Regional specialization and organization. You have generally a hierarchy of neurons from just a few on up to several thousand. The representations are built up by integrating information from similar inputs, and they can be complicated phenomena like word representation. The next, word representation, we're going to delve a little more into that now. That slide is not, let's skip over it. Okay. Functional magnetic imaging, fMRI, is a way that you can see the activity of neurons when they're still in situ and in living patients. The insert with the brain sections has red and yellow little clusters on it that represent activity in the neurons, and that activity has been recorded by specialized magnetic resonance probes that indicate when the neurons are active, namely when they're firing and conducting an action potential. And they function in word representation in response to words. They're found more extensive activation in the left hemispheres compared to the right hemisphere. But bilateral activity exerted when clusters of words were presented in a narrative or other context tended to be more bilateral. Next slide. Additional brain areas are involved in written language, visual form areas, VWFA, the parietal cortex and the occipital cortex. These are just examples of where the impulses are integrated and represented in representations. Next slide. On the left here, you see some specific brain regions that are that form the area for the virtual word recognition. And they're in a section of the temporal cortex called the fusiform gyrus. And its function is recognizes written letters and words and has a connection to brain areas processing visual information, links meaning to written language. So here you're incorporating visual cues, namely the writing in the text that you have before you, and you're trying to convey input to previous words that you've learned, sometimes verbally, sometimes by previous written language. The next slide. The parietal cortex and occipitotemporal lobe work together to identify shapes of written numbers or symbols. They connect them to their concepts and build meaning through constructing hierarchical connected representations of concepts. So you have multiple hierarchies that each represent a particular visual field, and the fields depend on fluid and efficient retrieval and manipulation of semantic knowledge. And the parietal and the occipital cortex are part of the processing procedures. And the parietal cortex receives input from the occipital, which I believe are part of these colored sections right here in the lower left-hand portion of the slide. And the occipital lobe offers input to the parietal cortex. Next slide. Agnosia is the various defects that individuals have. They're damage to the brain, resulting in problems with recognizing and identifying visual stimuli. Agnosia is particularly confined to visual stimuli. Usually is due to damage in the temporal lobes, which is where a lot of those visual stimuli are processed, and it causes different forms of agnosia depending on where the damage is in the temporal lobe. DBO was a 72-year-old man who suffered multiple strokes, and while he was able to see, he was unable to correctly name the objects in front of him. And we had a preceding slide that described his malady a little bit. Next slide. Another area is the fusiform area, which recognizes the recognition of faces. Damage to this region results in a disease called prosopagnosia, whatever it is, prosopagnosia, and face blindness. You don't recognize individuals by their complex faces. Next slide. The parahippocampal place area is the PPA, and gives rise to landmark agnosia. Its function is to respond to specific locations, pictures of buildings, particular scenes, or sequence of inanimate objects. And the damage to this particular area, the PPA, results in a failure to integrate any prior knowledge of those scenes into the representation that you're comparing the new input into. So the new input doesn't go anywhere. Next slide. Language processing. The language processing occurs primarily in the temporal lobe. Speech sounds and individual work involve the left and right temporal lobe. Anterior temporal lobe possibly participates in sentence level comprehension. And it produces complex speech dependent on the posterior temporal lobe and frontal cortex. And so the representations involve both those parts of the brain. Next slide. Visual and motor areas. Speech comprehension may require input from visual areas. And for analyzing letters and words, and output then occurs in the motor areas, example, controlling of the hands in conjunction with the words and letters that you see. Next slide. Insight into language research. The FOXP2 gene is an example of a protein that is a so-called regulatory protein and turns other genes on and off in a variety of cells, including neurons. So the FOXP2 gene is known to have a nucleotide sequence, and if it's mutated in that sequence, the full regulatory functions of the FOXP2 gene don't work, and you end up with difficulty making mouth and jaw movements in sequences for speech, disrupting written and spoken language by affecting signaling in the dorsal striatum, and disrupts the flow of information through the striatum, resulting in deficits. That's a very wide range of outcomes from just one gene that is mutated and changes the format for its regulation. Next slide. The striatodorsal segment of the brain contributes directly to decision making, action selection, and initiation through the integration of the sensory motor, cognitive, and motivational, emotional information within specific corticostriatal circuits involving discrete regions of the striatum. So this is another example of the way in which the representations come together and are recognized by the brain in processing the information that we get. The sensory motor circuit also is in the left temporal lobe, important for producing complex speech, and its function helps communication between systems for speech recognition and speech production, and it's involved in speech development. Effects of brain damage, again, aphasia, disorder that results from damage to portions of the brain that are responsible for language, affecting how one communicates. We're going to talk about two small areas of the brain here, just as examples. The Broca form and the Livornia form. In the Livornia form, all the patient could say was one word, tan, and damage occurred to the part of the frontal lobe causing aphasia. In another patient, a different post anatomic analysis indicated that a different position of aphasia was occurring, and non-fluent aphasia because speech production is impaired, but comprehension is intact. And the damage was in the left frontal lobe. We'll see if we've got the next slide. Does it may show this? No, it doesn't. Let's go back. And it gives rise to slow halting in speech and required great effort and often lacks complex word or sentence structure. And comprehension is still intact, but understanding complex sentences may be poor. And now we'll go on to the next example. Patient SA who had damage to their Wernicke area and Symptoms: difficulty comprehending speech, but not producing it. The speech may be normal in tone and speed, but it's riddled with errors in sound and word selection, and it's usually unintelligible. So those are just a few examples of particular patients with very, very specific deficits, very pronounced deficits that allowed us to get a handle on the regions of the brain that were affected. Next slide, please. Pure word deafness is a type of aphasia where the patient is unable to link sounds of words to their meaning. It generally involves the superior temporal lobes, which are indicated on the slide in the image on the right. And they can detect emotion, tone, and sex of the speaker, but the individual with that deficit cannot hear speech, music, or other sounds, and can still make sense of written language because visual information bypasses damaged auditory comprehension in the temporal area. Now, we're moving on up. When we get to executive function, we're talking about very complicated behavior. Let's see what the next slide shows. Executive function processes occur in the prefrontal cortex primarily, and they're gradual and more apparent in children. Activity decreases in children and adults as they mature. You may have recalled a little finger touching and fist formation. The child was told to do the opposite of the adult showing them, and a 3-year-old had great difficulty doing that task, but a 4-year-old didn't have much problem with it. So that was a maturing process in the executive function ability. Taking in sensory and emotional information and using this information to plan and execute decisions and actions is a part of executive function. And they select, rehearse, and monitor information being retrieved from long-term memory. Next slide. The prefrontal cortex, which is where this occurs, is spelled out in a little greater detail in this slide on the right. In interaction with a large network of posterior cortical areas and encodes specific types of information. And executive functions occur here. And neurons in the prefrontal cortex keep information active or in mind while carrying out a task that depends on it. Next slide. The prefrontal cortex also has the ability to suppress a behavior or action when it is inappropriate. It's called an inhibition stage, and it develops with age. The next slide shows the working memory, another core information processing of the executive function in the prefrontal cortex. It has the ability to maintain and manipulate information or a rule while deciding how to act over a brief period of time without external aids or cues. And you can read the rest of that. And the third phase is shifting or mental flexibility, which allows one to adjust their ongoing behavior when conditions require it. Error monitoring is more evident in adolescence, and by the time an adolescent reaches age 30, they have better control of this function, and they can maintain their executive functions in a more normal state. Decision making requires a person to weigh values, understand rules, plan for the future, and make predictions about the types of outcomes of choices. This is perhaps one of the higher functions of the brain. It's found in logical reasoning, emotional consequences, and it may result in the selection of logical and rational considerations as opposed to more emotional consequences that are found in mature brain. Next slide. This also involves the prefrontal cortex. Lateral portion of the prefrontal cortex is especially important in overriding emotional responses. The last part to mature occurs in one's early 20s. Strong connections to regions related to motivation and emotion are typical of the prefrontal cortex, with the inputs from the amygdala and the nucleus accumbens, which exert top-down control over emotional and impulsive responses. The next slide. The orbital frontal cortex is important in situations involving reward and punishment. And it's associated also with addiction and social behavior. Finally, in social neuroscience, we get to the presumably the highest level of brain function. It's still somewhat controversial. Social neuroscience is defined as the study of neural functions that underlie interpersonal behavior, so between persons. Large parts of the brain are dedicated to processing information about other people, include parts of the medial and lateral prefrontal cortex. And examples are reading social cues, understanding social rules, choosing socially appropriate responses, and understanding oneself and others. Next slide. Next slide. Mentalizing underlies empathy and theory of mind. Mental states of others usually understood through observation of their actions, and the brain must see and recognize others' movements and facial expressions to draw reference about their feelings. It also allows us to organize judgment about ourselves and judgment about others. Next slide. And mirror neurons are thought to be a component of that. They were initially identified in primates, and their function is to fire when performing specific actions, but also when watching another individual perform that same action. And they're controversial due to little direct evidence supporting their role in the theory of mind, mentalizing, and empathy, but there is some speculation mirror neurons may underlie our ability to understand another person's actions. And I think that's the concluding slide of this chapter.
Yes, it is. And I'm going to stop this share and I can pull up our next PowerPoint.
We're a few minutes before 6, and I told Gabby that the chapter 5 was by far the most complicated of the 4 that I had to discuss. So I should be able to get through these in faster time.
Fabulous. Before we move on, are there any questions?
I don't think so. I'm good.
I'm good.
Okay. Let me share my screen, if it will let me. Share screen. Share. Can everyone, oh, it's doing the weird thing again. Let's see, new share. I think I have to stop and do a new one. I appear to be having some technical difficulties. Does not want to populate. Okay, can everyone see this?
Yes.
Alrighty.
So this chapter is on the way that the brain develops from an early embryo on through a born child. And so let's take a few of these slides and see if we can form the concepts. The development of neurons during embryonic growth. Signaling molecules turn on certain genes and turn off others. So this is not just the geographic proximity of neurons to each other, but it's their chemical environment that is constantly under revision. Cell division or proliferation, pools of early stage brain cells increase by billions, and migration, newly formed neurons travel to their destinations. All this is going on at the same time. Signaling molecules are present, the cells are proliferating and migrating. Next slide. Next slide. So this is an early embryo here. It already has developed 3 areas, an ectoderm, the outermost layer, the mesoderm, the middle layer, and the endoderm, the innermost layer. Next slide. And as the cell begins to develop, the blastocyst basically was what I showed in the previous slide. And various stages of the fetus are shown in the lower row. Cells in each layer contain identical DNA instructions for development, giving rise to a variety of tissue types making up the human body. And the diversity of signals produced in those surrounding tissues are what turn certain genes off and other genes on. Yes, I think we can go on. The fate of a developing cell is determined by its proximity to various sources of signaling molecules. The concentration of each signaling molecule decreases farther from its source and creates gradients throughout the brain. And let's see if it gives an example of that in Sonic Hedgehog. Yes. Sonic Hedgehog is secreted from mesodermal tissue. So that's the middle layer beneath the developing spinal cord. As its exposure to signal adjacent nerve cells are converted into a specialized class of glia. Cells further away are exposed to lower concentrations of secreted Sonic Hedgehog, causing them to become motor neurons that control movements of muscles. And at even lower concentration, Sonic Hedgehog promotes the formation of interneurons, which relay messages to other neurons rather than to muscles. So that's a rather dramatic example of a single molecule having different effects depending on its concentration in the gradient. Proliferation. Nerve cells and progenitor cells arise from a small pool of neural stem cells and progenitor cells. Progenitor cells are special cells that can divide and become a variety of mature cell types. And you can see neural progenitors going on to neurons on the right part of the slide, and glial progenitors going on to form astrocytes and oligodendrocytes. Next slide. Before achieving their cell fate, mature cell fate, pools of cells undergo a series of divisions, which ultimately increases cells that form in the brain. Early divisions are symmetric, resulting in 2 daughter cells that are identical. And as the divisions progress, cells divide asymmetrically, giving ultimately rise to 1 daughter cell that can keep proliferating, and the second daughter cell progresses toward its ultimate cell fate as a neural or glial cell. This has clinical significance in that protein defects can cause a premature switch in the occurrence of symmetric and asymmetric division. The proliferative process permits rapid cell growth during early development. Billions of cells are produced in a matter of weeks. And after the original series of divisions is complete, only a few neural stem cells and progenitor cells remain within the brain. like those involved with memory. Next. This slide points out microencephaly and megaloencephaly. Brain that is reduced in size is the microencephaly, resulting in severe neurological deficits. And megaloencephaly is abnormally large brain associated with various neurodevelopmental complications and due to excessive cell proliferation. Migration. After neural induction and proliferation occurs 3 or 4 weeks after the baby is conceived, new neurons move from the inner surface of the embryonic brain to long-term locations. The ectoderm begins thickening and building up along the midline of the embryo. Next. And as cell division continues, a flat neural plate grows. Parallel ridges form arising from either side of the midline. Ridges extend from the head end along the length of the embryo. And a few days later, the ridges fold towards each other and fuse into a hollow tube. And that is essentially the progenitor of our spinal cord. And the head end of the tube thickens into 3 bulges: the hindbrain, the midbrain, and the forebrain. At week 7, the first signs of eyes and brain hemispheres appear. And I don't think I need to read the points 5 through 7. You can read them yourself. Guidance mechanisms. The most common guidance mechanism are radioglia, responsible for 90% of migration. They project radially from the intermediate zone to the cortex. And that's the first time I've seen this slide. It's fairly detailed. Neurons use these glia as scaffolding and inch along these glia to reach their destination. The process of radial migration occurs in an inside-out manner, and a different mechanism allows neurons to migrate sideways or tangentially. And that's just the way the guidance of the neurons occurs. No need to memorize specifics on this slide. So I won't go over it. You see a 6-step process. Clinical significance are intellectual disabilities or epilepsy. If migration is affected negatively in some way, due to alcohol, drugs, radiation, which may prevent proper migration and cause misplacement of cells, and also due to mutations in genes that regulate cell migration, like that sonic hedgehog was one example. Yes. Next slide. Making connections. Okay. After neurons reach their final destination, they begin making connections. These determine how particular functions like vision or hearing can occur. Induction, proliferation, and migration can occur internally during fetal development, while making connections depend increasingly on external afterbirth experience. Neurons become integrated through their branches of dendrites and long axons. And all of this architecture is being determined without any sensory input. That's important to know. Next slide. Axons and growth cones. Growth cones are ending in axons are ending in a growth cone, and they can span many times the size of their cell body. Growth cone is an enlargement at the tip of the axon that actively explores the environment to seek out its precise destination. Molecular cues guide it, as shown. Molecular cues in the growth cone. Some stud the surface of cells or are secreted into areas near the growth cone. Receptors on the growth cone enable its response to environmental cues. And binding of environmental molecules tells the growth cone to move forward, stop, recoil, or to change direction. And there's attractive, repellent, and signaling molecules that convey this. Next slide. Introduction to synapse formation. Once axons reach their targets, specialized connections called synapses begin to form. Because the brain contains millions of synapses, it contains millions of neurons, and it contains even more millions of synapses, it has a large capacity for information processing. At the synapse, only a tiny space separates the signaling portion of the axon from the receiving portion of the dendrite, and chemical cues can either promote or hinder the generation of a new electrical signal in the receiving neurons. The combined effects of cues from thousands of synapses determines how a receiving neuron responds. So all of those features that we went over in chapter 5 took for granted the fact that the brain already existed in a complex form. Next slide. The formation of synaptic connections must be highly specific. Some specificity is due to the mechanism that guide each axon to its proper target. Additional molecules mediate target recognition when the axon reaches the proper location. Dendrites are also involved in initiating contact with axons. Both sides produce proteins spanning the space between them and anchor synapses together. Next slide. Synaptic differentiation. Once contact is established, the synapse continues to differentiate. The presynaptic side, which is a tiny axon terminal that contacts the dendrite and becomes specialized for producing neurotransmitters, and the postsynaptic side, where the presynaptic neuron makes contact with the postsynaptic cell. Stocks itself with neurotransmitter contacts and proteins, enabling those packets to be held in place and then released. On the postsynaptic side, receptors respond to neurotransmitters from the presynaptic side, and both sides ensure synapses can transmit signals quickly and effectively. Next slide. In addition to neurons, there are other cells in the nervous system. One type of cell is called an astrocyte. It's a type of glial cell, and its functions are thought to only provide scaffolding and passive support to the neurons in the early stages of research. That turned out to be a false assumption, and we now know that they actually exert their own influence on synaptic development and function, especially since one astrocyte can contact thousands of synapses across multiple neurons, and molecules secreted by astrophytes regulate the synaptic development. Next slide. Neurotransmitters. An array of signals determines which neurotransmitter a neuron will use to communicate. Examples, those neurons that have developed into motor neurons generally have only a single type of neurotransmitter in the postsynaptic cell, acetylcholine. The type of neurotransmitter is not fixed for all neurons. Example, immature neurons with no other cell types produce norepinephrine on their own. And when cultured with other cell types like cardiac tissue, the same neurons produce acetylcholine. Genetic and environmental signals can modulate development of specialized cells and neurotransmitters. Signals to engage the gene and final determination of the neurotransmitter is influenced by factors that come from the location of the synapse itself. And at this point, I will say that the number of neurotransmitters that we're familiar with is small, among very simple molecules, a dozen or a couple of dozen molecules. But some neurotransmitters are thought to be small peptides, and those can run into the hundreds or even thousands. Next slide. Molecules secreted by astrocytes may cause degradation of synapses that occur during aging. And the defects in molecules promoting proper synapse formation may result in autism. Next slide. Myelination. The other type of cell that was present besides neurons and astrocytes is oligodendrocytes, as shown in the previous slide. And these cells are highly specialized to develop the ensheathment of axons. The cartoon shown in the lower diagram is about 7 sections of ensheathment, and in between each section is just a few nanometers of this absent a myelin sheath, and we'll see what effect that has. Next slide. Myelination is the fatty wrapping of axons by extensions of glia increases 100 times the speed at which signals can travel along. So the myelination of a neuron results in up to 100 times faster speed of transmission. The myelination acts as an insulation that covers wires and preserves the strength of electrical impulses that travel through them, and formation of myelin occurs throughout one's lifespan. Next. Now, interrupting the sheath is gaps causing an increase in the speed at which signals travel along the myelin sheath due to how the sheath is wrapped. The alternating pattern of sheath and nodes allows electrical signals to move down the axon faster. This is because electrical signals jump from node to node. This process is called saltatory conduction and is responsible for the rapid transmission of electrical signals. Pairing back. After the initial growth, the neural network is paired back to create a more efficient system. Only about half of the neurons from development survive to function in adult life. Next. Apoptosis removes entire populations of neurons through programmed cell deaths initiated in the cells themselves. This process is activated if the neuron does not receive enough life-sustaining chemical signals called trophic factors, which are produced in limited quantities by target tissues, and each supports the survival of a distinct group of neurons. The clinical significance is that neurodegenerative disease kills neurons by activating their cell death programs. Next. Neurons also form too many connections and have to be pruned back as an individual matures. Due to the formation of too many connections, pruning occurs depending on relative activity of each connections. Connections that are active and generating electrical currents survive, and those with relatively little activity are lost. Next.
We may have reached.
The end.
Oh wait, no.
Okay.
Yeah. All right, next one.
End of chapter 6.
Are there any questions while I pull up the next slides?
I'm good.
Okay.
Yep.
Going to chapter 7 now.
Okay. All right, let's see if I can share this.
Oh, you're getting good. Jordan.
Thank you. All righty.
So we're now into the adolescent brain and the infant child leading up to adolescence as well. Why study early development? The first years of life. Let's go on to the next slide. After a baby is born, its brain is only 13 ounces, and development continues until in the adult it's 3 pounds. So it's quite a 5 or 6-fold increase. Next slide. The rate of brain growth after birth is 1% per day. Next. But by the time that the embryo reaches 3 months of age, it's down to 0.4% per day. It's 64% larger than at birth, and the number of neurons in the cortex increases by 20 to 30%. 3 months after birth, synaptogenesis, the formation of synapses, the points of contact where information is transmitted between neurons, and glial cells are also multiplying and providing myelination by oligodendrocytes. Next. 2 years after birth, the brain has reached 80% of its adult size, 50% more synapses than an adult brain, and its number of synapses increase rapidly during the first couple of years of life. In fact, too many synapses for the brain to maintain because they all use energy and resources. During early childhood, the brain reduces its synapses and fine-tunes connections, and synaptic pruning is based on a toddler's experience. Next. 5 years after birth, the brain has reached 90% of its adult size. Next. And the fastest growing brain regions are the cerebellum at the base of the brain, which more than doubles in its size. And this is the brain region associated with learning, motor skills, and movement. So it is a region with the most amount of neurons. Next. Experience shapes the brain. Next. Compared to other animals, humans are born with less developed brains that take longer to mature and develop more extensively after birth. This has the advantage that the human brain is more easily shaped by experience, allowing for adaptation to the surrounding environment. Next. Critical periods, the time during which a given behavior is especially susceptible to and indeed requires specific environmental influences to develop normally. There's examples here of inputs from sensory and motor and emotional aspects, and genes are working with the environment as strong influences in the formation of neural circuits, and changes in neural connections during critical periods coincide with high learning rates, like speaking language. Next. Into adolescence. Stronger connections beat weaker ones. We've already alluded to that previously. And extensive synaptic pruning and restructuring occurs. And we've referred to that also as well. The brain improves connections with neurons by extending their dendritic branches, and the myelination of axons increases, especially in the frontal lobes. Second critical period in which complex function of the brain develop is influenced by environment and experience. Next. The consequences of the developing brain are enhanced connections, changes in the brain's reward system, and changes in balance between frontal and limbic regions. These all can contribute to teenage behaviors like risk-taking, sensation-seeking, and aspects of enhanced learning ability. One consequence that's cited here is a risk of addiction. Drug use causes damage to memory, attention, and executive functioning. Longitudinal studies track subjects over extended periods of time, and they're significant because they reveal how one's early environment and life can affect outcomes later in life. Next. These are technical achievements that are available to clinicians. MRIs and DTIs show an increase in white matter volume, especially in the corpus callosum, and may explain enhanced learning capacity due to increased connections. Also, gray matter declines with age, but density increasing in the left temporal lobe until age 30. And fMRI measures the brain activity. Next, please. Different brain regions experience different rates of growth and maturation. Brains continue developing until around 30 years of age. Brain development in an individual's 20s shows change in where myelination occurs. Early life, there's more in the visual, auditory, and limbic cortices. After 30 years of age, there's more in frontal and parietal neocortices. Remember, these areas are important for working memory and high cognitive functions. Frontal lobes develop last and gain more myelin later in life. Plasticity. Plasticity is the ability of the brain to modify itself and adapt to environmental changes like sensory inputs. Plasticity is necessary for critical periods. It responds to the environment and experience. There are two types of plasticity: experience expectant and experience dependent. Next slide. The expectant plasticity integrates environmental stimuli into normal developmental matters. Exposure to common or universal environmental experiences during limited critical or sensitive period of development, such as seeing language, seeing faces, and being held. Next slide. Experience-dependent plasticity. It continues to change in organization and specialization of a person's brain regions due to life experience that is not universal or anticipated, and skills developing throughout life with no critical or optimal period. Occurs in adulthood, demonstrating that the brain is always changing in response to experience. Next. Two-photon imaging allows one to see living neurons and track their growth after various experiences. The example of that on the right, where a neuron has been transfected with green fluorescent protein, allowing you to image and in fact, to focus on various shapes of the brain that you can see microscopically. Is there another slide or not? I don't think so.
Nope. We have reached the end of this one. I think we have one more PowerPoint to go.
Is there any questions from anyone so far?
No, sir.
No, I'm fine.
Okay. Jordan, if you want to download the next chapter 8.
All right, I'm trying to load it now. It doesn't like me. Okay.
Oh, yes. It's liking you better all the time.
Yay.
This chapter is on the adult and aging brain. So we're past the infant stage and the adolescent stage. and we're on to the adult stage and aging of the adult stage. Next slide. In childhood and adolescence, the most important brain regions are organized with areas near each other, working together to accomplish a cognitive task. When we reach adulthood, the brain's organization is more widely distributed, with distant areas connected by these long axons and working together. The human brain loses gray matter through pruning neurons and synapses, slows down in one's early 20s. White matter increases in brain regions strength and connection, and major nerve tracts are wrapped in myelin, and the peak volume reaches in one's 40s. Next. The prefrontal cortex, most important part of the adult brain, is to wire up. Remember, this region is responsible for higher level cognitive abilities like planning, problem solving, and decision making. And it's also important for cognitive control because it includes the ability to suppress impulses in favor of more appropriate responses. This is important, particularly in adolescence. Next. Intelligence. Different cognitive abilities have different patterns of maturation. Intelligence peaks between 25 and 60 years. Fluid intelligence, namely solving problems and identifying patterns, peaks at around age 30 and declines with further age. And what's called crystallized intelligence, which is vocabulary and knowledge of facts, increases until about age 50. Next. What is aging? Next. Normal versus pathological aging. Next. And an example of pathological aging is dementia, which is a chronic, persistent disorder of the mental process caused by brain disease or injury and marked by memory disorders, personality changes, and impaired reasoning, interfering with a person's day-to-day function. Healthy aging, some mental health decline is normal. Subtle changes in brain structure, chemistry, and function normally begin in midlife. Understanding healthy aging requires a knowledge of lifestyle and the choices that we're offered in diet, exercise, relationships, stress, and so forth. Next. How the brain changes. Next. Next. Cognitive changes are focused in memory. Declarative memory declines with age. Remember, memory for autobiographical life events, namely episodic memory, and memory of learned knowledge, namely semantic memory. Non-declarative memory remains largely intact with age, and this includes procedural memory. We're not generally aware of procedural memory. Working memory is the ability to hold a piece of information in mind and manipulate it. And it is an example of fluid intelligence, a set of cognitive skills that depends on rapid processing of new information rather than on stored knowledge. And working memory declines with age. Next. Attention. The ability to focus on a particular stimulus and filter out distractions becomes more challenging with age. Next. The ability to focus on two tasks at the same time also becomes more challenging with age. Next. Structural changes that can occur. Changes in brain volume. The brain volume begins to decline in one's earlier 30s or 40s, and volume loss may not be uniform throughout. Some areas seem to shrink more and faster than other areas. And the biggest losers, which worsen with age, are the prefrontal cortex, cerebellum, and the hippocampus. Next. Changes include the shrinking neurons, retraction and decreased complexity of dendrites, and loss of myelin. Next. A second change is cortical thinning. The cerebral cortex thins with age. Cortical thinning similarly affects certain regions of the brain more than others, most pronounced in the frontal lobes and part of the temporal lobes. Next. In this region of the brain, they have the most pronounced declines in volume and cortical thickness with age. Remember, these areas took the longest to reach maturity, and the last in, first out theory of brain aging is consistent with the last parts of the brain to develop are the first to deteriorate. White matter and projection fibers support this hypothesis. Next. Neuronal changes. We get changes in synapses and dendrites. Synaptic changes are highly selective and subtle, but their effects are greater than those of structural and chemical changes. Dendrites shrink, their branches become less complex, and they lose dendritic spines with increasing age. Dendritic spines are tiny protuberances that receive chemical signals, generally from the axon, and may be invoked in working memory, and loss of these could impair neuronal communication and contribute to cognitive decline. Next. Neurogenesis is the formation of neuroneurons, new neurons. This neurogenesis declines with age, continues in those brain regions previously believed to halt after birth. The olfactory bulbs, the factory bulbs, and the dentate gyrus of the hippocampus. The rate of neurogenesis in humans experiences a modest decline with age, and strategies boosting neurogenesis can enhance cognitive functions. Next. Chemical changes. Next. 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 Oxidative stress is one reason that the brain ages. It's caused by an imbalance between production and accumulation of oxygen reactive species, ROS, in cells and tissues, and the ability of a biological system to detoxify these products. This imbalance can be seen in individuals with Alzheimer's disease and other cognitive impairments like memory. Next. DNA damage over a lifetime may contribute to the aging process throughout the brain and body. Damages include base modification, abasic sites, and strand breaks. May be caused by oxidative stress. Next. Mitochondria and free radicals. An organelle in every cell of one's body that functions as a powerhouse and carries out chemical reaction that provides energy for cell use. That's the mitochondria. Some metabolic reactions produce harmful byproducts known as free radicals, which are highly reactive molecules, which, if left unchecked, can destroy fats and proteins vital to normal cell function and damage DNA. And defense mechanism to neutralize these free radicals declines with age and leaves the brain more vulnerable. Metabolic activity in the brain demands around 20% of the body's fuel. It makes the brain more vulnerable to metabolic changes. As the brain's energy demand remains high, its energy supply can no longer keep pace, and the brain's ability to take up and use glucose diminishes and mitochondrial metabolism declines. Next. Immune dysfunction often occurs in conjunction with metabolic changes. The functions of the brain's immune cells, which are present largely in the microglia, defends against pathogens, cleans up cellular debris, and maintains and remodels synapses. Inflammatory responses are protective, but prolonged states are harmful. Next. This is the microglia that I mentioned in the previous slide. Glial cells derived from mesoderm that function as macrophages in the central nervous system. It has an important role in brain infections and inflammations, and it becomes more reactive with age. So microglia are yet another type of cell in addition to neurons, astrocytes, and oligodendrocytes. Impaired protein recycling. Next. Normally, cells break down and recycle damaged proteins and molecules. Over time, damaged molecules can build up in cells and prevent them from functioning normally. Neurons in the brain are not replaced as often as cells in other parts of the body, so they are more vulnerable to a buildup of damaged molecules. Cellular breakdown and recycling process declines with age, reducing the efficiency of waste removal systems. Next. All this has a clinical significance. Excessive buildup in abnormal proteins in the brain contributes to age-related neurodegenerative diseases, including Alzheimer's and Parkinson's. Buildup of proteins and other cell components can contribute to cellular degeneration. And changes in the brain due to aging can occur within the context of other changes throughout the body. For example, worsening cardiovascular health could contribute to or drive changes in the aging brain. And healthy aging. Diet and exercise. Poor cardiovascular health puts a patient at risk of age-related cognitive impairment, as we noted in the last slide, and the development of dementia. Diets rich in vegetables, fruits, and whole grains, but low in meals and dairy products reduce risk factors. Why? The reduction of risk in high blood pressure and high levels of LDL cholesterol. Next. Specific nutrients may be linked to improving cognitive performance and lower rates of dementia. These nutrients include antioxidants, vitamin C and E, and flavonoids and omega-3 fatty acids. Does this mean we should consume copious amounts of these nutrients? Why or why not? Let's see if it's answered in the next slide. Yes. Reducing the number of calories eaten without leading to malnutrition can lead to an improved cognitive health and a longer lifespan. How many calories are in a gram of carbohydrates, a gram of protein, a gram of fat? Next. Exercise. Aerobic exercise can improve cognitive function and offset some declines from aging. Engaging in regular physical activity promotes improved learning, improved memory, reduced risk of developing dementia, reduced cortical thinking, and less shrinking of the hippocampus. Aerobic exercise may also slow the progression of Alzheimer's disease and dementia and increase neurogenesis, enhancing neuroplasticity. Exercise also improves neuroplasticity, possibly through increasing neurogenesis. Remember, neuroplasticity in the brain's ability to form and recognize connections between neurons and responses to changes in behavior and environment. Mental stimulation and social cognitive stimulation, mental stimulation and large cortical improvements can also improve cognitive function in aging. These include engaging in cognitively demanding work, stimulating activities, and having an active social life. This cognitive stimulation also appears to enhance neuroplasticity by increasing neurogenesis and by boosting levels of important neurotrophic factors. Next. If there is another. Okay, that's the end of our slides.
You've reached the end. Does anyone have any questions?
I just have one when it comes to nutrition. You kind of hear the buzzword of probiotics can help reduced brain functioning, what's the newest research that you've heard or have worked with in regards to using probiotics for brain health?
Well, that's controversial.
Okay.
You'll find advocates and people who are skeptical on both sides of the issue. I think small amounts of supplements are relatively good if they are antioxidants, for example. But there's a limit to the amount of them that the human body can process, and that result in any enhanced cognition.
Okay. Thank you.
So, all of these slides, I believe Gabby sent them along to the teachers, so everyone should have access to them. And a recording of this lecture will be sent out later this week by Gabby.
Okay.
Are there any more questions from our student?
No, I'm good. Thank you.
Okay, well, if you do come up with a question when reviewing, please know that you're always welcome to reach out to us with the questions. We're so excited to have you, and we're so excited to have Dr. Rosenberry speaking to us today. Thank you so much for doing that. You did a wonderful job, Dr. Rosenberry.
Yes,
you.
thank you. Thank you very much, Doctor.
Thank
Okay.
you.
I'm not sure if Gabby is currently on with us. It looks like she has stepped away. I know she's in the middle of some experiments.
Oh, fun.
Yes. But I'm going to say good night and
Okay.
session if that's good with everyone.
Yes,
Okay.
it is.
Thank
Thank you
you.
very much.
So much. See you all next month.
Okay.